Good day, everyone. Today we'll be discussing the fourth of the four major biomolecules we'll be discussing during the biochemistry, no? So, this one is a pretty lengthy lecture kasi meron na rin ako mga sinama ditong processes that are important for your knowledge as a medical technologist, as well as medyo mahaba kasi talaga siya. So, please grab a coffee, grab a snack. You can actually pause this video anytime you want. First, since this is a recording, and feel free to go over it over and over again para mas understand nyo yung topic. Kasi this is very fun and a very high yield topic for the future. Kasi makaka-relate kayo dito. So, next. Here are the objectives for today's lecture. Huwag na nating daanan yan. Let's go ahead. So, before we begin the discussion on nucleic acids and the structure niya, yung mga pagkakaiba ng DNA and RNA, I want to walk you through what you call the central dogma of molecular biology. So, the central dogma of molecular biology is a sequence that describes the flow of information inside your cell. So, itong si central dogma, nung na-discover to, is actually a very monumental discovery. No? So, what is the central dogma then? So, the central dogma describes the flow of information from DNA to RNA to protein. So sabi natin, di ba, your cells needs to make proteins in order to perform its function. So how do you make your proteins? First, you'll go from the DNA. So your DNA is transcribed into RNA, then translated into the protein that is required. So the DNA is actually the one that is carrying the instructions for protein synthesis. But in order to make your proteins from the DNA instruction, you have to first convert it to RNA in order to produce your protein. So, your DNA can also be replicated. So, pwede siya ma-duplicate para, for example, di ba, mag-reproduce yung cell mo, gagawa ka ng another copy of the cells during cell division. Siyempre, replicate mo yung DNA. So, DNA, when you make another copy of a DNA, you call the process replication. Okay? When your DNA is converted to RNA, in order to have a template for protein synthesis, you call the process transcription. When your RNA naman is used as a template in order to guide your protein synthesis, you call the process translation. Kasi parang magkaibang lengguwahe siya, you converted it, from example, from English to Filipino, di ba? Kaya parang ganun yung nangyayari dito. So, from RNA, gagawin mo siyang protein. So DNA to DNA, replication. DNA to RNA, transcription. RNA to protein, that's your translation. So, itong reverse transcription, discuss natin siya later on. But in general, the flow of information is this one. So, DNA is converted to RNA, which is also converted to protein during in your central dogma. Okay? Next. So, doon na tayo sa building blocks of your nucleic acids, no? Kasi sabi nga natin, di ba, pag nagdi-discuss naman tayo ever since una pa lang, when we discuss these topics, we start with the building blocks. And the building blocks of your nucleic acids are your nucleotides. So, your nucleotides are the building blocks of your nucleic acids. So, nucleic acids yung topic natin. So, why are they called nucleic acids? So, bakit nga ba siya naging nucleic acids? Kasi, nung na-discover siya, ni Friedrich Miescher during the 1980s, he was studying the nucleus of the WBC. So, dahil nakita siya sa nucleus, ginamitan siya ng nucleic na prefix, and then dahil nakita niya na acidic tong molecules na to, they call it a nucleic acid. So, naging nucleic acid yung tawag sa kanya. And the monomers for these nucleic acids are your nucleotides. So, let's look at the building blocks of your nucleic acids. So, yung nucleic acids natin, yung nucleotides natin rather, yung building blocks, are actually made up of three components. You have a pentose sugar, you have a phosphate, and you have a base. So, yun yung tandaan yung three parts of your nucleotides. You have a phosphate sugar, you have a pentose sugar rather, you have a phosphate, and you have a base. So, tandaan nyo that the pentose sugar is the one that your phosphate and base is bonded to. So, the pentose sugar is bonded to both the phosphate group and the nitrogen-containing heterocyclic base. So, nitrogen-containing heterocyclic base or your nitrogenous bases. So, yung sugar na yun, there are two types na pwede siya. Pwedeng ribose, pwede rin siyang deoxyribose. At ito yung nagde-determine kung siya ay magiging DNA or RNA. Pag DNA, the sugars are deoxyribose. Kapag RNA, the sugars are ribose. Okay? Next, meron siyang contained phosphate. And then it also contains a nitrogen-containing heterocyclic group or tinatawag natin nitrogenous bases. So there are two types of nitrogenous bases. You have your pyrimidines, you have your purines. So pag pyrimidines, it's the six-membered ring. Kapag purines, it's a fusion of a five-membered and a six-membered ring. So you have a fusion of a five-membered ring and a six-membered ring. So pag six-membered ring lang, it's a pyrimidine. Pag 5 plus 6, that's purine. So, ano naman tong magkakaibang, so, we have two purines, we have three pyrimidines. So, ano tong mga purines and pyrimidines na sinasabi natin? You have purines which are adenine and guanine, or represented by the letters A and G, respectively. And then sa pyrimidines naman, you have your thymine, cytosine, and uracil. So, thymine is represented, of course, by letter C. Cytosine is is represented by letter C. And then uracil is represented by letter U. So, tandaan nyo, both purines are actually found in both DNA and RNA. Okay? Meanwhile, your pyrimidines, depende kung DNA or RNA kung ano yung makikita mo sa kanya. In DNA, you can find both thymine and cytosine. In RNA, you can find cytosine and uracil. Walang thymine. Okay? So, in DNA, you find cytosine and thymine. Walang uracil. In RNA, you find cytosine and uracil. Walang thymine. So, how do you remember kung sino yung purines? Sino yung pyrimidines? You remember the term pure Ag. So, pure Ag is pure silver. Ag, silver, di ba? Yun sa periodic table, yung symbol ng silver is Ag. So, pure silver, pure Ag, that's purine. Pwede nyo rin gawing pure as gold. So, mas partial ako dun sa pure as gold kasi Ag, isipin ko pa na silver. So, pure, so, hindi ko naman kailangan yun. So, pure Ag, pure as gold, pure silver, whatever, floats your boat, you can do whatever you want. So, mnemonic lang yan. So, purines, Ag, pyrimidines is CUT. Okay, so cut. Okay. Next. So, tapos na tayo dun sa nitrogenous bases natin, no? So, now let's go to what you call so how do you form your nucleotides? Let's discuss this. How is it formed? Before you form your nucleotides, you form something called the nucleoside. So, your nucleoside is the fusion of your pentose sugar plus nitrogenous base. Kasi diba sabi natin yung nucleotides are made up of three. Meron siyang pentose sugar, meron siyang nitrogenous base, meron siyang phosphate. So, dito sa nucleoside formation, wala pa yung phosphate. Ang pagsasamahin mo pa lang is your pentose plus your nitrogenous base. So, convention lang before we go into it, di ba? When numbering your carbons or your atoms on your bases, you use 1, 2, 3, 4, 5, 6, whatever. Kung ilang atoms yung meron dyan, okay? 1, 2, 3, 4, 5, 6, 7, 8, 9. Yun yung numbering dito sa nucleoside. Ay, sa nitrogenous base. When you are numbering naman your carbons on your sugar, you add a prime. So, prime yung i-add mo kapag you're talking about sugar. So, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So, pag sugar, you add a prime. Kapag dun sa nitrogenous base ka ng number, you don't put a prime. Okay? Clear? Let's go. So, your pentose sugar plus nitrogenous base, you form your nucleoside. So, O-side, tandaan nyo, may Os. So, you're in the involved one is the sugar plus the nitrogenous base. Okay? So, what happens? A condensation reaction happens. That's what. So, we have a sugar. And then you have your nitrogenous base. Ano yung nagko-condense? Diba? Pag may condensation ka, you form water. Diba? Pag malamig yung baso, magkakaroon ng condensation sa labas. Magkakaroon ng water. So, condensation, you form water. So, yung water, by forming that water, yung N dito, yung N, on the 9th position will bind to the C1. So the nitrogenous base will bind to the C1 prime of your ribose molecule. So when the condensation occurs, your nitrogenous base is attached at C1 prime, and then you form your nucleoside. So yun yung nangyayari. So in nucleoside formation, you form a nucleoside and one molecule of water. So how do you name your nucleosides? You name them based on what nitrogenous base is involved. So, kapag you're talking about purines, the suffix is osin. If you're talking about pyrimidines, the suffix is actually idine. So, for example, yung purines natin, diba? Sabi natin, purines are pure as gold. So, pure Ag, pure silver. So, si purines is adenine and guanine. So, pag purine, sabi natin, the suffix is osin. So, this one will become adenosine. This one will become guanosine. If the sugar involved is a ribose, wala kang prefix. If the molecule involved is a deoxyribose, you add the prefix deoxy. So, kapag ito, ang sugar involved is a deoxyribose, then it becomes deoxy, adenosine, deoxy, guanosine. Okay? If you have your pyrimidines naman, your pyrimidines are your CUT, di ba? Sabi natin, cytosine, uracil, and thymine. So, it's cytidine, uridine, and thymidine if the sugar involved is ribose. Kapag deoxy naman, it's deoxycytidine, deoxythymidine, deoxyuridine. Okay? So, that's how you name your nucleosides. So, after you add the pentose sugar plus the nitrogenous base, syempre may kulang pang isa, which is your phosphate group. So, dun tayo sa phosphate group. How do you add it? You add it during nucleotide formation by attaching the phosphate to the C5' molecule on the sugar. Kasi nga, di ba sabi natin, your phosphate group and the nitrogenous base are both attached to the sugar. So, sa C1' ang naka-attach is the nitrogenous base. Sa C5' ang naka-attach is your phosphate. So, the phosphate is attached to C5' using another condensation reaction to form what you call a phosphoester linkage. So, para siyang ester sa carbon, pero phospho yung nakalagay. So, that's a phosphoester linkage. And the reaction occurs by a condensation wherein yung H tsaka OH will form water and then this P will attach to this O. So, magkakaroon ka ng phosphoester group. Phosphoester linkage dyan. So, how do you name this then? You have your nucleoside and then you add a 5' monophosphate to the nucleoside name. So, this one is the nucleoside, diba? Sabi natin, sugar plus nitrogenous base, that's a nucleoside. So, nucleoside plus phosphate, monophosphate, pag isa lang, diphosphate, pag dalawa, and then triphosphate if tatlo sila. So, run down lang kung paano naming, di ba? Since we mentioned it earlier, if you're talking about your purines, then the suffix is osin. If you're talking about your purines, the suffix is osin. Kapag RNA siya, walang prefix. Kapag DNA siya, may deoxy. If you're talking about pyrimidines, like your cytosine and thymine and your uracil, then the suffix is idine, idine, idine. If it's a ribose, then walang prefix. If it's a deoxyribose, then you put the prefix deoxy. And then, if nagkaroon na siya ng phosphate group, you add the suffix or you append 5' monophosphate pag isa lang, diphosphate pag dalawa, triphosphate pag tatlo. Okay? Next. So, if you have a sugar plus a base, you form the nucleoside. Kapag meron na siyang phosphate, that's when you start having your nucleotide. So, kapag isa lang yung phosphate, you add the monophosphate. Pag dalawa yung phosphate, you add a diphosphate. Pag tatlo yung phosphate, then that's the triphosphate. Okay? Clear? Next. So, how do these nucleotides form chains to form your nucleic acids? So, para lang siyang actually yung similar to your proteins, no? Meron siyang backbone, tapos doon sa backbone na yun, may mga nakalawit na yung mga R group ng proteins natin, no? So, in a similar manner, dito sa nucleic acids, you have a backbone of alternating phosphate, sugar, phosphate, sugar, phosphate, sugar, di ba? So, alternating phosphate, sugar, phosphate, sugar, phosphate, sugar. So, depende na lang kung ano yung sugar mo, that's a DNA or an RNA. Kapag deoxyribose, syempre, that's a DNA. Kapag ribose, syempre, that's an RNA. And then, ang mga burloloy, kunyari, dun sa sinabi natin, di ba, para siyang charm bracelet or para siyang Christmas lights, no? So, yung mga nakasabit dun sa banderitas mo, basically, yung pise is the backbone. And then yung mga nakasabit is your bases. So, the bases protrude from the chain at regular intervals, nakakadikit sila sa sugars. Okay? And then, how do you differentiate the various DNAs and RNAs together? Siyempre, you look at the variable part of the molecule, which is the bases. So, the backbone is consistently a phosphate sugar, phosphate sugar, phosphate sugar na alternating. And then nakasabi sa kanila yung mga nitrogenous base. Okay? So, sabi natin kapag deoxyribose yung sugar, that's DNA. Kapag ribose yung sugar, then that's an RNA. So, what are the other differences between your DNA and RNA? Here they are. Let's discuss them. So, your DNA is deoxyribonucleic acid. Your RNA is ribonucleic acid. The sugar is deoxyribose for DNA. The sugar is ribose for RNA. You have the same three bases, adenine, cytosine, guanine. For DNA, adenine, cytosine, guanine. For RNA, pareha silang merong adenine, cytosine, guanine. Ang pinagkaiba nila sa DNA, yung thymine, you have thymine. Sa RNA, you have uracil. So, you have, sa DNA, you have thymine. Sa RNA, you have uracil. So, your DNA is also mostly found in the nucleus, while your RNA is mostly found in the cytoplasm. So, syempre, magkaiba sila ng function, magkaiba sila ng structure, magkaiba sila ng location, magkaiba rin sila ng function. So, your DNA is for the storage and transfer of genetic information. It indirectly controls the function of the cell, and it is the one that is passed on from existing to new cells during cell division. Your RNA naman serves as a template for the synthesis of your proteins, and it is synthesized from DNA during translation. So, sabi na nga natin yun sa central dogma, diba? DNA to RNA is translation, then translation of your RNA to proteins is your, DNA to, synthesized from DNA from transcription to rather. That's not translation. So, synthesized from DNA from during transcription. So, your DNA to RNA, that's transcription. RNA to protein, that's translation. Okay? Next. So, so let's discuss the primary structure of your nucleic acids. So, that the structure in which the nucleotides are linked together. So, para siyang primary structure din ng proteins, diba? So, primary structure ng proteins, that's the amino acid sequence. Dito naman sa nucleic acids, the primary structure, of course, is also the sequence of your nucleotides. So, meron din siyang, it's very similar to your proteins na meron siyang directionality. Kung sa proteins, di ba, you have the N-terminal and the C-terminal. Dito sa primary nucleic acid structure, you have the 5' end and the 3' end. So, sa 5' end, nandun yung phosphate. Sa 3' end, nandun yung free OH ng sugar mo. So, the 5' end has the free phosphate at the C5'. Kaya siya prime end, di ba? Kasi di ba C5', di ba may mga prime tayo doon pag number tayo nung mga sugars natin, di ba? So sugars, when you number them 1 2 3 4 5 6 7 prime, you add a prime. So 5' end, nandoon siya sa C5' na carbon. May naka-attach na phosphate. And then on the 3' end, so sa C3' may naka-attach na OH. So yun yung 5' to 3' na direction. So when you read your bases, you go from 5' to 3'. So from this one is the 5', then the direction is this way. Pag magbabasa ka, for example, this is your sequence. Okay? So sabi na natin, the primary nucleic acid structure is the sequence in which the nucleotides are linked together. So, tandaan nyo, meron siyang directionality, as mentioned, from 5' to 3', yung pagbasa sa kanya. Next, tandaan that each non-terminal phosphate group has a 1- charge. So, dahil nga, itong phosphate groups na to, actually come from phosphoric acid. So, kaya siya nagiging acidic, kaya siya naging nucleic acid is because of the acidic behavior, because of the free meron siyang phosphoric acid. So yung O na yung PO4 na yan, yung phosphoric acid na yan, originally meron siyang tatlong OH groups. So yung dalawa nasasama siya sa linkage ng ating nucleic acid structure. And then the remaining OH group, nag-donate siya ng H. So yung H na yun, kaya siya nagiging acidic. Kasi diba H is acid. So, H plus is acid. So, yung H plus na yun, kaya siya naging acid. Kaya siya naging nucleic acid. Okay? Next. The non-terminal phosphate groups are bonded to sugars via 3' 4' phosphodiester bond. So, anong ibig sabihin nun? So, meron kang, here is a ribose molecule. So, here is a phosphate. So sabi niya, meron tayong 5' phosphodiester bond. That's not 4. So, 3', 5' phosphodiester bond. So, ano nangyayari dito? You have a 5'. Ito yung, di ba? Sabi natin, 5' is the 5th carbon dun sa ribose mo. And then, in another amino acid, so this one is another one is another nucleotide, di ba? Ito bagong nucleotide. Doon sa carbon, doon sa ribose niya, siyempre 3' doon sa ribose na yun. Dito sa sugar na to rather. So 3' no sugar na yun, merong is bonded to a phosphate. And then this phosphate is bonded to the 5' group on the next sugar. Ulitin natin para mas madaling maintindihan. Meron kang 3', 5' phosphodiester linkage. Okay? So, you have a sugar. It is bonded to the next nucleic acid rather, by a 3' on that sugar, bonded to a phosphate, and then that phosphate is bonded to the 5'. So, pag ganun siya, napa-zigzag. So, 3' phosphate, the phosphate is bonded to 5'. The 3' is bonded to the phosphate is bonded to the 5'. So, the 3' is bonded to another phosphate, which is bonded to another 5'. Kaya siya nagiging phosphodiester bond. Magkakaroon ka ng dalawang esters. Okay? Phosphodiester bond. Kasi meron kang phosphoester and another ester. So, that's a diester bond. You have an ester, that's a diester bond. Phosphodiester bond, and a phosphodiester bond. So, 3', 5' phosphodiester bond. Okay? So, edit na nga natin ito para hindi na tayo malito. So, that's a 3', 5' phosphodiester bond. Okay? Next. Doon tayo sa ating next discussion, which is, para lang mas madali nyo lang ma-visualize, no? Meron siyang in common with the protein primary structure. Meron siyang directionality, as mentioned earlier, di ba? Directionality ng protein natin, merong N-terminal end at meron kang N-terminal end at meron kang C-terminal end, and it goes this way. Okay? So, N-terminal to C-terminal, yun yung directionality sa protein natin. Meanwhile, sa nucleic acid, you have the 5' to the 3' end, and you read it this way. So, another thing in common is that it has backbone that does not vary in structure. So, ito yung protein backbone mo, the alternating amide plus alpha carbon plus amide plus alpha carbon plus amide plus alpha carbon. Meanwhile, yung sa nucleic acid structure natin, ang alternating is the phosphate, then sugar, phosphate, sugar, phosphate, sugar. Diba? Sabi natin. And then, another thing in common with your proteins is that the sequence of attachments to the backbone is the distinguishing feature. Kasi nga, yun yung variable characteristic ng mga nucleic acids natin. Diba? Ang nagbabago sa protein mo is the R groups. Ang nagbabago naman dito sa nucleic acid mo is what? Nitrogenous base is attached to the sugar. So, ganun yung analogy ng ating protein structure with the protein primary structure with the nucleic acid primary structure. Okay? So, run down ulit nung diniscuss natin kanina, no? You have a nucleic acid structure, diba? Your nucleotide is made up of a sugar, a phosphate, and a base. The base plus sugar is a nucleoside. The base plus phosphate plus the sugar is a nucleotide. So, you put your nucleotides together, you form your nucleic acid. And there are two types of your nucleic acid. You have a deoxyribonucleic acid and a ribonucleic acid. So, the bases are same for the most part, except you have thymine for deoxyribonucleic acid. You have your uracil for ribonucleic acid. Okay? Next. let's go to the discussion of your DNA molecule. So your DNA molecule uh, is the storage form of genetic information in our cell, no? So how was the DNA discovered? It's actually very controversial tong topic nito, no? So the discovery of the DNA structure, of course, started with Friedrich Miescher, as we mentioned earlier. Siya yung naka-discover, di ba, ng nucleic acids. Siya yung in-isolate niya yung nucleic acids, and he called it nuclein. So, in 1868. So, sinaunang panahon pa si Friedrich Miescher nung na-discover niya yung nucleic acid. Well, syempre, of course, as time went by, ano yung mga scientists add on to his discovery. So, pero hindi pa nila alam at the time what that nuclein is actually for, di ba? Basta alam lang nila merong certain something found in the nucleus and they call it a nucleic acid. Hindi nila alam na that nucleic acid is the one that actually carries the genetic material of the cell. So it wasn't until Oswald Avery, Colin MacLeod, and Maclean McCarthy produced compelling evidence na nagkaroon sila ng idea na that your nucleic acids actually carry your genetic information, no? So, ang ginawa kasi nitong tatlong to is they extracted a virulent strain of Streptococcus pneumoniae, an important bacteria that causes pneumonia, and used it to make a non-virulent strain to a virulent strain. So, parang ang ginawa nila is kumuha sila ng strain ng Strep pneumoniae, ginamit niya yung ginawa niyang pathogenic yung ginawa niyang pathogenic yung other non-pathogenic strain ng Strep pneumoniae. So, yung ginamit nila dun sa experiment nila, na-demonstrate nila na it's actually the DNA from the virulent strain that is responsible for the virulence. So, na-demonstrate nila that the DNA is the one that carries the genetic information from virulence. So, and then after that, you have Alfred Hershey and Martha Chase, which wasn't mentioned here. Pero what they studied were bacteriophages. Tapos doon na lang nalaman na it is actually your DNA that carries genetic information. So another important clue to the structure of your DNA was postulated by a man named Erwin Chargaff and his team, no? So, hindi lang sa Erwin Chargaff, meron siya mga teams, pero si Erwin Chargaff siya kasi yung leader. So, yung mga iba, et al. Lang sila sa research. So, ang main author talaga is si Erwin Chargaff. So, si Erwin Chargaff and his colleagues in the 1940s came up with what we call now as the Chargaff's rules. So, meron tayong 4 basic Chargaff's rules. Ito yung 4 basic Chargaff's rules. You have the first one is that the base composition of DNA varies from one species to another. Next, the DNA from different tissues have the same base composition. So, kung in base composition, by base here, we refer to the nitrogenous bases. So, nitrogenous bases. So, DNA from different tissues have the same base composition. So yung DNA mo, sa pita mo, sa batok mo, sa puso mo, sa utak mo, pare-parehas lang sila ng base composition. Kasi, yun na naman yung DNA mo sa katawan mo, okay? Next, the base composition of a DNA in a given species does not change with an organism's age, nutritional state, or changing environment. So, yung DNA mo, when you're young, pag gutom ka, pag busog ka, pag tanda mo, pag pumunta ka sa desert, pumunta ka somewhere, hindi siya nagbabago. Okay? So, the base composition of your DNA does not change with an organism's age, nutritional state, or changing environment. Okay? Next. So, in all cellular DNAs, the number of adenosine is equal to the number of thymidine residues, and the number of guanosine residues is equal to the number of cytosine residues. So, ano to? Adenine is equal to thymidine. Guanosine is equal to cytidine. Okay? With adenine is equal to thymidine. Guanosine is equal to cytidine. So, adenine is equal to thymidine. Guanosine is equal to cytidine. So, diba, tandaan nyo, your purines are your AG. Your TC is your pyrimidines, diba? So, tandaan nyo, the number of purines is equal to it follows that the sum of the purines is equal to the sum of the pyrimidines. So, pag nag-add mo sila, since parehas lang naman itong dalawa, and then parehas lang itong dalawa, if you add them, then the purines and the pyrimidines are equal. Okay? If you sum it up. So, this relationship is what you call the Chargaff's rules. Okay? Next. Another thing in the discovery of the structure of the DNA is itong very controversial rule, itong very controversial for scientists na to. You have Rosalind Franklin and Maurice Wilkins as well as James Watson and Francis Crick. So, ano nangyari dito anong issue sa kanila, diba? So, Rosalind Franklin, siya yung primary researcher actually. She used X-ray diffraction to photograph and analyze DNA. So, ito yung photograph niya ng DNA molecule, no? So, nung pinicturean niya yung DNA na yan, they call this photo as a photograph 51. So, makikita mo kasi na meron siyang para magkakaroon ka ng idea that your DNA is a helical molecule. No? So doon na realize na DNA is actually helical. So, si Rosalind Franklin and si Maurice Wilkins yung nakuha ng photographic evidence that the DNA is a helical molecule. Pero, si James Watson and Francis Crick are actually the ones credited for deducing the structure of the DNA and postulating a 3D model that accounted for all of the available data at the time. So if tinanong ka sa discovery ng DNA double helix, kung sino yung nag-propose ng structure ng DNA double helix, you answer James Watson and Francis Crick. Pero yung talagang nakahanap ng evidence regarding the structure is Rosalind Franklin and Maurice Wilkins. So, anong chika dito? Si James Watson and Francis Crick, allegedly, kinuha nila yung data ni Rosalind Franklin ng walang paalam. So, allegedly, si Maurice Wilkins yung kumuha ng data, tapos binigay niya kay James Watson and Francis Crick. So, ano nangyari? Nagkaroon sila ng hint na helical nga yung DNA molecule. So, pero syempre, they also did a lot of the work. Kaya sila nakagawa ng DNA model. Kasi syempre, hindi mo naman ma-deduce yung base pairing, etc. ganyan. But just by looking at one photo. So, meron din silang, syempre, meron din silang contribution. But it's not just all Rosalind Franklin dahil siya yung nakapicture ganun. So, Rosalind Franklin is the one that took the picture along with Maurice Wilkins. And then James Watson and Francis Crick, sila yung nag-deduce ng structure sa pagtanong ka. So actually, James Watson and Francis Crick, as well as Maurice Wilkins, all won Nobel Prize for this. Unfortunately, Rosalind Franklin was already dead by the time they won the Nobel. So, hindi siya na-award. Kasi, hindi sila nag-award ng posthumous na Nobel Prize. So, in theory, dapat may Nobel Prize din si Rosalind Franklin because she's the one who actually took the photo. Okay? So, doon na tayo sa structure ng DNA double helix. Diba? So, diba sabi natin double helix yung structure na na-postulate ni Watson and Crick. So, paano siya naging double helix? You have two polynucleotide strands coiled around each other in a manner that is similar to a spiral staircase. So, para ka sa spiral staircase, tapos yung mga steps mo, yun yung binding between the nitrogenous bases. So, the sugar phosphate backbone, siya yung strip or kung sa spiral staircase, siya yung railings, di ba? So, spiral staircase mo, siya yung railings. This one is a backbone. This one is another backbone. And then, in between the two strands, you have the bases extending inward between them. So, dito, sa base na to, ito yung mga naka-extend na. Dito sa backbone ito yung mga naka-extend na base. Then, on the other backbone, ito naman yung mga naka-extend na base. So, yung mga bases nila, magharap sila sa isa't isa, magkaganyan. Okay? And then, imagine mo, this is the backbone here. And then, the bases are in between. So, ganyan. So, this ko na, for example, this is guanine, this is cytosine, this is adenine, this is thymine. So, magkaganyan sila. Tapat-tapat. Okay? Next. And then, tandaan nyo that the DNA double helix is actually anti-parallel. Bakit sinabi nating anti-parallel? Kasi when you read them, one strand is going downwards from 5' to 3'. The other parallel strand is going from 5' to 3' pero pataas. That's why it's an anti-parallel. 5' to 3', 5' to 3'. So, anti-parallel yung direction niya. Parallel sila pero magkasalungat. So, they are connected to So the antiparallel strands are connected by hydrogen bonds. So hydrogen bonds between the complementary bases. So ano yung complementary bases mo? Adenine is complementary to thymine. Cytosine is complementary to guanine. So ito yung cytosine, complementary to guanine, and between them is a hydrogen bond. Are hydrogen bonds. Adenine is complementary to thymine and between them are hydrogen bonds. Ganun din dito. So, marami kang hydrogen bonds in between the nitrogenous bases linking the two DNA strands together. So, tandaan, your DNA double helix is like a spiral staircase. Yung backbones form the railings. Yung steps in between is the bases. And the bases are made up of your nitrogenous bases linked together by hydrogen bond. Okay? Sorry, I'm sick no, so please forgive me if I sniff from time to time or sneeze. Okay. So next, dun sa tinatawag nating base pairing. Kasi di ba sabi natin, your DNA double helix is anti-parallel. Tapos in between them, you have the nitrogenous bases that are complementary to each other. So meron kang nitrogenous bases on this strand. Meron kang nitrogenous bases on this other strand. So ganyan siya. Diba? They are complementary. So, ganyan siya. Pero syempre, dahil you want to form a straight helix, you want to form a straight molecule, there is a limited size in between the double helix. And ang kasya lang doon actually is a pairing with one small base, between one small base and one large base. So, yung one more large base natin, syempre, siya yung pyrimidine. Yung merong 5 plus 6 na helical structure, di ba? And the one large base is the purine. So, baliktad. Yung small base mo is your purine, the large base is your pyrimidine. So, magkabaliktad na to. So, you have one large base, pyrimidine, and then one small base na purine. So, correct lang natin, ha? The small base is the purine, the large base is the pyrimidine. So, your structure, the interior of the double helix can only accommodate one small plus one large. So, dito sa one small base mo is a purine, and then one large base is a pyrimidine. So, yun lang yung kasya na sa base pairing. Pero bakit A bonds with D? Bakit G bonds with C? Bakit hindi pwedeng A bonds with C or G bonds with T, di ba? Kasi mas favorable yung AT sa GC, kaya siya nangyayari. Bakit? Kasi, as you can see here, mas favorable yung binding ng adenine thymine keta sa thymine guanine because of the structure, di ba? Kasi dito, if you have a thymine and an adenine, you can form two hydrogen bonds. Pero pag thymine guanine yung ginawa mong pairing, then you can only form one hydrogen bond. So hindi siya masyadong malakas yung kapit, di ba? Meanwhile, if you have a cytosine adenine, isang hydrogen bond na yung magagawa. Pero pag cytosine guanine, mas marami kang hydrogen bond magagawa. Makakatatlo ka pa nga eh. So, mas favorable yung adenine thymine base pairing and cytosine guanine base pairing compared to the thymine guanine and cytosine adenine base pairing respectively. So, sa molecules natin, sa DNA molecules natin, syempre doon tayo sa mas favorable. So, mas nakikita tong thymine adenine cytosine guanine na base pairing. So, and because these base pairings are very predictable, na pag may thymine ka dyan, yung partner niya is adenine. Pag may cytosine ka dyan, malamang ang partner niya is guanine. Dahil alam mo yung pagiging complementary nila na property, you are able to predict what the opposite strand is. Okay? So, let's have some exercise kung paano tayo mag-predict ng opposite strand. No? So, here is your predicting the base sequence of a complementary strand. Diba? Sabi natin, adenine is complementary to thymine. Guanine is complementary to cytosine. So, ito, we have a given strand here. You want to predict the sequence of bases on the complementary DNA strand. Sabi natin, the direction is parallel. So, anti-parallel, 5' to 3' this way. Then, this other strand must be pag ganon sa pakabila. So, this one is the 5' end. This one is the 3' end, di ba? Okay? Next. So, dito, how do you predict the complementary strand now, di ba? Tatandaan nyo to. Complementary strand, di ba? So, if you have a C, the complement is letter G. If you have a G, the complement is letter C. If you have an A, the complement is letter T. If you have an A, the complement is another letter T. If you have a letter T, the complement is letter A. If you have a C, the complement is letter G. And then this one is another G. And this one is a letter A. And this one is a letter T. Okay? So, tandaan, ang direction is 5' to 3', pero since the given is already 5' to 3', then the complementary strand must, dito sa leftmost, malamang 3' yon. So, dahil siya ay anti-parallel. If you want to write it in 5' to 3' form, you just flip the orders. Magiging T, A, G, G, A, T, T, C, G, C, 3' siya. If you want to see it in 5' to 3' na tamang order. Pero mas maganda if when you're predicting the complementary strand, you do it this way. Okay? Next. So, what are the interactions that stabilize your DNA double helix? Dalawa lang naman sila. Pero dahil napakadami nila, it becomes very strong. Okay? So, the first one is your hydrogen bonding interactions. Diba sabi nga natin, the link between your nitrogenous bases are, diba sabi natin, parang siyang ladder. Doon sa ladder mo. So, parang siyang ladder. Doon sa ladder mo, yung mga steps niya is your nitrogenous bases. And then, doon sa nitrogenous bases na yun, the bonds between them are hydrogen bonds. So, hydrogen bonds by themselves are weak forces. Pero by virtue of the number of how many base pairings are there in a DNA molecule, nagiging very strong yung bonding ng DNA molecule. Kasi sobrang daming hydrogen bonds nga in between. Okay? Next, another interaction is the base stacking interaction. So, ito naman yung sa structure naman ng DNA double helix natin. So, the base stacking interaction is because of the positioning of the bases. So, the bases in a double helix are positioned with the planes of their rings parallel. Kasi di ba ang mga rings natin, di ba you have your pyrimidines and your pyrimidines ring, tapos naka-parallel sila. So, merong interactions between the bases above and the bases below a certain base. Diba? So, meron siyang interaction with the bases above it, meron siyang interaction with the bases below it. So, yung bases na yun, they interact with each other, and then nagsastack siya. So, meron kang hydrogen bonds, di ba? Stabilizing your double helix this way. And then you also have interactions stabilizing your double helix by the bonding of your bases to other bases above and below. Okay, next. Okay? So, and the type of interaction actually found in your between your purines and pyrimidines are hydrophobic interactions. Kasi yung purines and pyrimidines mo are hydrophobic in nature. Okay? So, now let's go to the process of creating your DNA. So, let's go to your DNA replication. So, DNA replication, may DNA ka, tapos sinerox mo. You make a identical copy of your DNA. The process is called replication. So, the DNA replication is the biochemical process by which DNA molecules are identical copies of themselves. Or meron kang gumawa ng duplicate ng themselves. So, paano nangyayari to? And what is the key idea behind your DNA replication? Then, well, the key idea is still your complementary base pairing. Okay? So, dito na tayo sa first part. Replication of your DNA. You use DNA to make another DNA. So, how does DNA replication occur? Diba sabi natin, you have a DNA double helix. So, para kang may ladder, tapos tinwist mo siya, or meron kang spiral staircase, that's a DNA double helix. So, you want to replicate that, of course, you have to unravel your DNA. So, how do you unravel your DNA? First, you separate the two strands of DNA, and then each strand will act as a template. So, dahil may template ka, by virtue of complementary base pairing, pwede mong gawin yung separate strand. You can use this strand as a basis in order to make the other strand. Okay? So, so each strand acts as a template for the synthesis of a complementary strand, and it results in two DNA molecules with bases identical to those of the parent. Kasi nga, dahil sa complementary base pairing mo, itong side na to, you can use this one. You can use this strand. Tapos ito, mapo-form mo yung complementary strand niya. Kasi gano'n lang naman yun. So, so you have a DNA, you split it, and then each strand serves as a template in order to make the complementary strand. Okay? So when you split it, each strand, so one strand serve as the template, and then you make a complementary strand. So magkakaroon ka ngayon ng dalawang DNA molecule. So each of your DNA molecule is made up of one old strand, kasi di ba hinati mo nga to, tapos kumuha ka lang ng complement. And then, itong other half naman, ginawa mo din yung complement niya. So, you have one old strand and you have one new strand. That's why you call the process of DNA replication as semi-conservative. Kasi semi-conservative, you conserve one of the strands and then the other strand is new. Okay? So, one strand is conserved, the other strand is new. So, and then, hold it in. So, ginagawa if you want more copies. Okay? So, how does DNA replication occur? The first step, yung pag-unwind ng DNA natin into two is acted upon the influence of an enzyme called helicase. And when you split your DNA double helix, diba, parang siyang ladder, pag binuka ka mong ganun, yung tawag doon sa part where it is unwinded is the replication fork. Okay? So, and then, when you split your DNA, yung ma-expose na ngayon yung bases of each strand, no? And then, you can add new nucleotides to that strand. Add new nucleotides to that strand using your DNA polymerase. Okay? So, paano ba nangyayari to? So, gawin lang natin siya para maintindihan nyo kung ano nangyayari sa DNA replication. So, this is your DNA. This is the parent DNA that you want to duplicate, diba? So, complementary base pairing, sabi natin, A is bonds with T, A bonds with T, G bonds with C. So, kita natin yung pagiging complementary base pairing niya, di ba? So, dito, sabi natin, your DNA is unwinded, and the enzyme responsible for that is helicase. So, your helicase will act on this DNA and produce what you call a replication fork. So, ito yung replication fork. So, ang mangyayari is bubuka ka yung DNA mo. Tapos, may expose yung nitrogenous bases. So, anong mangyayari dito? Then, you have nucleotides that go in and form the complementary strand to these exposed bases. So, let's look at this one by one. No? So, because complementary to A is letter T, complementary to C is letter G, complementary to G is letter C, complementary to C is letter G. So, this one is A, this one is another A, and then this one becomes T. So, yan yung mga magdidikit na nucleotides dyan. Meanwhile, dito naman sa baba, you have a T here, then yung nucleotide na mag-complementary base pair sa kanya, of course, is letter A. Kapag C yan, ang magdidikit sa kanya is letter G. Kapag G to, ang magdidikit sa kanya ay letter C. Kapag A to, ang didikit sa kanya ay letter T. And then pag A to, ang didikit sa kanya is letter T. And then this last one, syempre letter T siya, so ang didikit sa kanya ay letter A. So if you look at the sequence, di ba? If you look na ito yung ginamit mong template, then you generate the complement. Then you produce a daughter strand. You produce another daughter strand. So, tignan mo ngayon yung itsura ng daughter strand mo. AGC-TTA, TCG, AAT. AGC-TTA, TCG, AAT. AGC-TTA, TCG, AAT. So, yung daughter strand mo is identical to each other and it's identical to the parent DNA, diba? So, itong part na to is from the parent. Itong part na to is a new. So, di ba, sabi natin, DNA replication is semi-conservative. So, semi-conservative replication of your DNA. You split a parent DNA. You use the strands as the template. in order to produce new strands. Okay, and then you have two daughter DNAs that are identical to each other and to the parent DNA. Okay, so itong picture naman ito sa side is how you add your nucleotides. So, for example, this one is the parent strand, this one is the growing chain. So, itong sabi natin complementary base pairing, di ba? So, T will bond with A, T will bond with A, G will bond with C, C will bond with G. And then your G, for example, dito sa example natin, you have a guanosine nucleotide here. This G will bond with letter C to form hydrogen bonds here. And then, of course, the phosphate phosphodiester bond here. Diba? Phosphate will bind to the 3 prime, 5 prime on the 2 riboses above and below it. Okay? Next. So, your DNA replication actually usually occurs at multiple sites within a molecule. Hindi siya yung may isang part lang yan, tapos aantayin mong matapos na yung buong haba. No, it can occur in multiple sites between a molecule, and it can actually occur bidirectionally. So, the site at which your DNA replication starts is what you call the origin of replication. So, merong certain base sequence found dun sa origin of replication that is recognized by proteins. And then, dun sa site na yon, they will deposit helicase. So, pag nakapag-deposit na yung helicase, then, gagawa na siya ngayon ng bubble doon. Gagawa na siya ng parang bubble. And then, of course, itong mga may araw na to, that's a replication fork. So, that's a replication fork. This one is another replication fork. This one is another replication fork. Okay? Next. So, ito na, zoom in natin yung pangyayari during DNA replication. So, dahil na-discuss na natin yung paano base pairing nangyayari, gawin na natin, pakita na natin yung machinery on what happens, what is used by the cell in order to duplicate your DNA, no? So, you have a DNA, you unwind it using your helicase. Your helicase breaks the H hydrogen bonds that hold the base pairs together. Di ba sabi natin, your DNA is like stairs, tapos yung steps niya is your bases, tapos nakatwist siya, parang spiral staircase yung structure niya. So, ang nangyayari dito is, you have a helicase that breaks the hydrogen bonds in between to unwind the DNA double helix at the replication fork. Okay? So, pag ibukan niya na yung helicase mo, naibukan niya na yung DNA molecule, para siyang zipper actually. Kasi di ba yung zipper, pag hinatak mo pa ganun, bubuka ng bubuka. Di ba? Pag meron kang zipper, tapos meron kang, ang ginagawa ng zipper mo is, binubuksan niya yung bag mo. So, pag meron kang zipper, tapos hinatak mo siya, lalaki ng lalaki yung gap in between, di ba? So, ganun yung nangyayari actually sa helicase. Para siyang zipper for your DNA molecule. Imagine the zipper, the length of the zipper is your helicase, tapos yung hinahawakan mo doon sa zipper, yun yung helicase. Siya yung nagbubukas ng zipper. So, the helicase will break the H bonds in between. And then, you have something you call the single-stranded binding protein. So, it will bond to a single strand on this side, tapos other single-stranded binding proteins will bond to a strand on this side. So, what does the single-strand binding protein do? It maintains separation of the strands. Kasi ang tendency niyan, kasi syempre, dahil may hydrogen bonds in between, tendency niya is mag-close. So, what maintains the separation? It's single-stranded binding protein. Next, you have another enzyme called topoisomerase. So, topoisomerase naman is located dun sa part ng DNA na hindi pa naka-wind. So, ang ginagawa ni topoisomerase is, nire-relax niya yung DNA kasi di ba double helix siya. So, ang ginagawa ni topoisomerase is, nire-relax niya yung DNA para maunat. Kasi pag unat siya, pwedeng, mas madali siyang buksan ni helicase mo. So, di ba, kapag yung zipper mo naman, nakapulupot, hindi mo naman siya mabubuksan na madali. So, ang gagawin mo, si topoisomerase, inuunat niya yung DNA, it relieves torsional strain, para pag si helicase is dadaan doon sa part na yun, madali niya nang ma-unwind yung DNA. Okay? So, next. So, what happens? You make a replication fork. Ang bubukas ng replication fork, si helicase. So, pag nakabukas na siya, may expose na si 5' end then si 3' end. So what happens during replication? You have what you call a primase. So primase will insert a short RNA segment to initiate replication. Hindi pa kasi yung enzyme needed for replication does not cannot add new bases without a primer. So una, your primase will add a primer, itong kulay yellow. For example here, the primer is added by a primase. So ito si primase, nag-add siya ng RNA primer here. So dito, may primase din na nag-add ng primer nito. Okay? So, pag may primer na, pwede nang mag-act ngayon yung main enzyme for DNA replication, which is DNA polymerase. Gumagawa kasi siya ng polymers. And then, the molecule involved is DNA. So, DNA polymerase will recognize the primers and add new deoxyribonucleotides kasi we're talking about DNA here. So, you will add new deoxyribonucleotides at the 3' end of the primer. So, 5' dito, 3' to. So, this one is the 3' end of the primer. Dito, mag-a-add si DNA polymerase ng more nucleotides. So, ang synthesis, dito siya mag-a-add hanggang humaba ng humaba, resulting in elongation of the strand in the 5' to 3' direction. So, this is very important, no? So, tandaan nyo that your DNA polymerase can only ever act in the 5' to 3' direction. Okay? Okay? 5' to 3' direction lang yung direction of movement ni DNA polymerase. Okay? So, magsa-start siya sa 3' ng primer. Didikit siya sa 3' ng primer and move in a 5' to 3' direction. Okay? So, good yun kung yung strand mo is going towards the helicase. Okay? Going towards the center of the replication fork. Kasi kapag yung strand mo na sinisynthesize mo is going towards the replication fork, habang binubuka ni helicase to, tuloy-tuloy siyang nagsisynthesize. Kasi 5' to 3', yung 3' mo nasa may fork banda. Pero kung yung 5' mo naman is yung nasa may fork, and then the 3' end mo is nandun sa may dulo ng molecule, then you have a problem. Kasi your DNA polymerase can only form molecules in the 5' to 3' direction. So what does it do? Ang nangyayari is nagiging discontinuous yung synthesis nung polynucleotide mo. So, kaya meron tayong tinatawag na leading strand and lagging strand. So, ano yung leading strand? Leading strand, ito yung strand na continuous yung synthesis. Kasi 5' to 3' direction, yung synthesis niya, yung 3' is malapit sa replication fork. So yung end nung nucleic acid niya nandito, tapos yung 3' end niya is nandito sa papuntang sa replication fork. Pero kapag yung 5' end mo is on this end, tapos ang 3' end mo is nandito, yung sinisynthesize mo, you form what you call your Okazaki fragments. Kasi ang ginagawa dito sa lagging strand natin is, you add a primer. Siyempre, sabi natin, DNA polymerase can attack without a primer. You add a primer, and then your DNA polymerase will act on that. So, hahaba siya ng pagganon until it encounters another chain. And then, pag nagawa niya na yan, you add another primer, tapos yung elongation will add on another chain. So, parang siyang tumatalon-talon, balik-balik. So, for example, dito, primer to, mag-synthesize siya that way. Pag tapos na siya dyan, dito naman siya ulit, kung saan nadagdag yung primer, and then, dun pupunta siya pa ganon. And then, you add another one here, and then you add it in that way. So, patalon-talon siya. From pag ganon, dito, pag ganon, and then, pag ganon, and then, pag ganon. So, hindi siya continuous na synthesis. Okay? Unlike dito sa leading strand, yung synthesis mo from 5' to 3' is tuloy-tuloy lang. Dito, para siyang nagtatahi na pabalik-balik. Okay? Next. So, here is the next step naman. No? So, nakagawa ka na ng strand mo. What you have now is your another thing I forgot to mention naman pala is your DNA polymerase has a proofreading activity. Meron siyang tinatawag na 3', 5' exonuclease activity. Exonuclease kasi pwede siyang magtanggal ng mga maling lagay. So, tatanggalin niya yung nung nucleotide doon na mali, tapos ah parang back-read niya, tapos mali pala, so tatanggalin niya yung mali, tapos lalagyan niya ng bagong tama. So si DNA polymerase, aside from being responsible for elongation, is actually uh, helpful in maintaining the integrity of your DNA strand kasi it has proofreading activity. Okay? So next. So you have another DNA polymerase. You have naman your DNA polymerase 1. So DNA polymerase 1 is another polymerase. So yung kanina si DNA polymerase 3, nilagay ko ba na 3 siya? So let's edit lang this. Let's edit lang this quickly. So DNA polymerase 3 is the one that recognizes the RNA primer and adds nucleotides and proofreads. Meanwhile, you have your DNA polymerase 1. So DNA polymerase 1 naman removes the RNA primers and fills it in with deoxyribonucleic acids. Kaya siya isang mahabang strip of DNA. Hindi siya DNA, tapos may RNA, tapos may DNA, tapos may RNA, DNA, tapos may RNA. Yung primers, tinatanggal din siya, tapos papalitan din siya ng deoxyribonucleic acids. Okay? And then deoxyribonucleotides. So ito, as you can see here, this is DNA polymerase 1. Kanina primer to, tinanggal niya yung primer, tapos nilagyan niya ng deoxyribonucleotides. So that's what your DNA polymerase 1 does. Next, so dahil this continues to, you have what you call a DNA ligase. So your DNA ligase will seal the nicks between your Okazaki fragments. So kung itong isang discontinuous strand na to, yung isang Okazaki fragment mo, hindi sila continuous with each other, then ang magsisil ng gap between them is the DNA ligase. How does it do that? By catalyzing the formation of your phosphodiester bonds. Kasi di ba sabi nga natin, the links between the nucleotides are your phosphodiester bonds. Okay. So, summary nung synthesis. Magkakaroon din tayo ng visualization nito. So, mas madali siya maintindihan. Summary of DNA synthesis. The DNA is unwinded by your DNA helicase to produce 2 template strands. And then, the nucleotides attached by hydrogen bonding and is joined by DNA polymerase. So, DNA polymerase. So, kapag continuous yung synthesis, you call that the leading strand. If the strand is made in short bursts, tapos you link them together, you call that lagging strand. So continuously, you add, that's the leading strand. If it's may Okazaki fragments, that's the lagging strand. So, and then the nicks between your Okazaki fragments are linked together by your DNA ligase. Okay, so I have a video here. I think this is very useful in visualizing what I was talking about just now. So let's play it. So the link is in the PowerPoint. So
DNA is a molecule made up of 2 strands, twisted around.
Sorry. Let's play it again.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay. So next, let's go to the organization of your DNA. So DNA is actually organized in condensed form of your DNA. Found in the nucleus is what you call chromatin. So chromatin is the condensed form of DNA that's why it can fit inside your nucleus. Kasi nga sabi nila pag in-unfold mo daw yung DNA mo, it's around 2 meters long yung isang DNA molecule. So paano siya magkakasya yung 2 meters dun sa ultra sa mga microscopic submicroscopic DNA molecules natin, di ba? So meron siyang arrangement, of course. So yung chromatin is described as a beads on a string. Bakit? Because of the structure. So discuss natin kung paano siya kinocondense, no? So, of course, you have your DNA. Your DNA is a double helix. So, your double helix DNA is wrapped 2 times around an octamer of molecules you call histones. Diba? Sabi na nga natin yun, diba? During the cell lecture, yung histones, yung DNA mo is wrapped around histones during the organization. So, ito yung histones. Itong bilog-bilog na yan. So, octamer dahil siya ay 8. So, meron kang 4. Tapos, meron ka pang 4 sa likod. Sa ilalim. Magiging octamer siya. So, para siyang cube. Nating 4. 4 above, 4 below. So, you have an octamer of proteins called histones. Tapos, 2 times iikot yung DNA doon to form a nucleosome. So, itong isang to is a nucleosome. This one is another nucleosome card. Okay? Next, another molecule called your histone H1 or your H1 histone binds to what you call the linker DNA. So, yung linker DNA yung nagli-link ng DNA molecule mo as well as your H1 histone. So, your H1 histone will bind to the linker DNA as well as your nucleosome core in order to stabilize the chromatin fiber. So, meron ka nucleosome core, linker DNA, histone H1, nucleosome core, linker DNA, histone. Meron kang nucleosome core, you have a linker DNA, H1, linker DNA, nucleosome. And then you have another H1, linker, nucleosome, linker, H1, linker, nucleosome, linker, H1, linker, nucleosome. So, ganun yung arrangement niya. Okay? So the binding allows it to stabilize in the chromatin fiber. Okay? So, itong ano na to, Ito itsura niyang ganyan. May H1, tapos may nucleosome, tapos may H1, tapos may nucleosome, tapos may mga linker DNA in between. Kaya siya tinawag, ito yung chromatin mo. That's why you refer to the chromatin as a beads on the string. Kasi mukha siya siyang string, tapos meron ka mga beads. No? So, yung beads mo, yung mga nucleosome core. So, okay, so, then this beads on a string, yung string na yan, is further compacted by winding into supercoils. Ang tawag naman dun sa supercoil na yun is your solenoid. So, I don't know if you ever had this experiment during your high school or kung nag-STEM kayo, high school or elementary, yung gumagawa ng electromagnet, di ba? If you have a nail, tapos babalutan mo siya ng alambre, and then yung alambre, idedikit mo sa battery, you form a magnet, tapos yung nail na may nakapulupot na alambre na yun, pag dinikit mo sa battery, magiging magnetic yung nail na yun. So, didikit dyan yung mga paperclip, etc. So, yun yung pang-alala ko dito. So, you call this thing a solenoid, yung nakapulupot na, naka-coil na ganyan. That's why this thing is also called a solenoid. Okay? So, this one is a solenoid of your chromatin. Next, let's go naman to your genes. So, kanina ba natin sinasabi, di ba? DNA is the storage form of information nung sa loob ng cell natin. So, ano ba yung i-store niya? Yung syempre yung mga genes natin. So, your genes is the fundamental unit of heredity. It encompasses the DNA sequences that encode the structural components of the gene product as well as those that regulate its expression. So, each gene in your body, syempre, meron siyang specific location. Kaya nga, and you call the specific location of a certain gene as the genetic locus. So, sa katawan natin, di ba, tandaan nyo, nung pinanganak tayo, nung ginawa tayo, ng mga magulang natin, di ba, you have a male and a female, they have sex, and then you form a zygote, and then, so, hindi tayo galing lang sa mother natin na nag-duplicate siya. Hindi tayo clones of our mothers. We're the combination of our father and our mother. So, sa humans, kasi we're made during sexual reproduction, some of our chromosomes are, half of our genetic material is from our mother, half of it is from our father. So, sa cells natin, yung mga chromosomes natin doon, meron tayong dalawang versions of each gene. Okay. So, in each version of the gene in our cells is called an allele. So, yung gene na yun, so sabi natin, di ba? Each version of a gene is called an allele. So, dahil meron tayong two versions for a gene, yung two versions na yun are actually found on the same genetic locus on the homologous chromosome. So, meron kang gene from your father, gene from your mother, yung gene sa chromosome 6 ni mother is homologous to the gene sa chromosome 6 ni father. Okay, so ganun yun. Meron silang counterpart. Meron sa mother, meron sa father. So for each gene, you have two alleles. One is from the mother and one is from the father. If pareha sila, you call it homozygous. If identical sila, then, if hindi sila parehas, then you call it heterozygous. Okay? So, hindi natin ko tatoksingin yung mga Punnett square, etc. So, tandaan nyo lang. One from the mother, one from the father. Kaya meron kang two versions of each gene. Okay? So, yung genome naman is different from your gene. So, the genome naman is the complete set of instructions. So, yung gene, yung specific sequence that encodes for a trait, si genome is the complete set. So, the complete set of genetic instructions found in an organism is the genome. So, sa humans, yung cells natin are diploid except for yung ploid is the suffix. So, pag haploid, it's only one set of genetic instructions. Kapag diploid, that's two sets of genetic instructions. So where do you get the two sets? One set is from your mother and the other set is from your father. Your sperm or egg, your sperm and egg are haploid cells. So haploid sperm plus haploid egg during fertilization combine to form your diploid zygote. So, yung mga cells ng katawan natin are diploid except for the sperm and egg. So, during fertilization, so during fertilization, you get 23 chromosomes. So, yung 23 yung one set, and then another set from your mother sa egg. So, 23, one set, 23, another set. So, the total, you have a total of 46 or 23 pairs of uh, 23 pairs of chromosomes. Okay? So, dun sa 23 pairs of chromosomes na yun, 22 pairs are actually, 22 pairs are actually autosomal. One pair is for the sex. So, autosomal, autosomal chromosomes, of course, responsible for all the traits of our body. The one responsible for your sex traits, of course, is your sex chromosomes. So, depende kung ano ipapasa sa'yo ng nanay mo, ipapasa sa'yo ng tatay mo, kung ano yung gender. So, yung isa mong X chromosome, always galing siya sa nanay. And then, your gender is determined kung ano yung ipapasa ng tatay mo sa'yo. So, kung ang pinasa ng tatay mo sa'yo is another X chromosome, then you become XX. So, pag XX ka, you are a female. Kapag ang pinasa naman sa'yo ng tatay mo is the Y, then you become an XY. So, pag XY ka, then you are a male. Okay? That's biologic, no? So, hindi yun kasama dun yung mga LGBTQIA+. Syempre, hindi na yun as far, we're talking about genetics, of course. So, pag XX, you're a female. Pag XY, you're a male. Okay?